Hey everyone, welcome to the Explainer. Today, we're jumping straight into 10 of those multiple choice questions from the recent A-level biology paper that might have left you scratching your head. We're not just going to find the answers, no way. We're going to break down the why behind them, really get into the biology, and seriously level up your exam game.
And I'm your exam coach for today. Look, my job isn't just to hand you the answers on a plate. It's to help you really understand the core concepts, so you can walk into that exam hall feeling confident, ready to take on whatever they throw your way.
So what do you say? You ready to get into the nitty-gritty of these 10 MCQs and really sharpen up that exam technique? I thought so. Let's get started.
All right, first up, we're diving into questions 11 to 15. This first block is all about biochemistry. So, we'll be looking at everything from the molecular bonds that give proteins their shape to how enzymes get to work.
Okay, question 11. Let's tackle this one together. We've got to figure out the interactions between these three pairs of R groups. First, look at X. See that OH group getting close to a COO group? You've got a slightly positive hydrogen and a slightly negative oxygen. That's a textbook hydrogen bond, for sure. Now let's look at Y. These R groups, they're just chains of carbons and hydrogens, totally nonpolar. And what do nonpolar things do in water? They huddle together, right? They want to get away from the water. That's your hydrophobic interaction. And finally, Z. This one's pretty clear-cut. You have a negative charge here and a positive charge there. Opposites attract. That's a strong ionic bond. So we've got hydrogen bond, hydrophobic interaction, ionic bond. That makes B our answer. Nailed it!
All right, let's check out question 12. Now, this looks like a standard enzyme graph, but there's a little trick here. The key is to notice we're measuring the amount of substrate that's left over. So, the question tells us the enzyme's happy place, its optimum pH, is around 1 to 2. What happens at the optimum pH? The enzyme works like crazy, right? It's at its fastest. So if it's working super fast, is it going to leave a lot of substrate behind? No way. It's going to use up the most substrate, meaning the amount remaining will be at its absolute lowest. Now, as you move away from that sweet spot, the enzyme starts to denature, the reaction slows right down, and, you guessed it, more substrate is left untouched. Which graph shows that? Graph A is the only one with that dip at pH 1 to 2. Perfect.
Okay, question 13. Time to talk about inhibitors, specifically non-competitive inhibitors. This is one of those questions where if you know your definitions cold, you're golden. So let's make sure we've got this down.
So, what's the deal with a non-competitive inhibitor? Think of it this way. It doesn't fight for the parking spot. That's the active site. Instead, it binds somewhere else on the enzyme at what we call an allosteric site. But when it binds, it kinda warps the whole enzyme shape. This messes up the active site, making it less effective. Because some of the enzymes are now basically out of commission, the overall maximum speed of the reaction, the Vmax, goes down. But here's the key part. Since it's not competing for the active site, the enzyme's actual attraction to the substrate, the Km, isn't affected. So Vmax decreases, Km stays the same. And that, my friends, is exactly what option C says.
Now for question 14. We've got two experiments here. The only difference is that experiment 2 has more enzyme. So, what does that mean? Imagine you have more workers on a production line. The work gets done faster, right? It's the same here. More enzyme means more active sites ready to go, so the reaction is going to be much faster. That means a steeper initial slope on the graph. But, and this is super important, both experiments started with the same amount of raw materials, the same amount of substrate. So in the end, they're both going to make the exact same amount of product. So, which graph shows a faster start but the same finish line? It's got to be graph C. Steeper curve, same plateau. Simple as that.
Okay, for question 15, the process of elimination is our best friend here. Let's go through these statements one by one. Statement 1, a protease can act as an antibody. No, that's mixed up. A foreign protein, like a protease, would act as an antigen, which triggers your body to make antibodies. So, statement 1 is out. What about statement 2? It says, adding more substrate can overcome a non-competitive inhibitor. Nope, we just talked about this. That trick only works for competitive inhibitors. So 2 is also wrong. That leaves us with statement 3. Water is needed to break peptide bonds by hydrolysis. Well, what do proteases do? They break down proteins. And how do they do it? By adding water to break the peptide bonds. That's literally the definition of hydrolysis. So statement 3 is 100% correct. And if only 3 is correct, our answer has to be D.
Hey, just a super quick break. If you're finding this useful and things are starting to click, you should definitely follow me over on Instagram and TikTok. I post daily revision tips to keep you on top of your game. Okay, let's get back to it.
And we're back. Time for our next block of questions, 16 to 20. We're switching gears a bit now, moving away from the molecular level and zooming into cell biology. Get ready for some membranes, transport, and a bit of mitosis.
Right, question 16. We've got a classic diagram of the cell surface membrane. Let's identify these molecules. Molecule 1 is a protein, and you see that branching chain coming off it? That's a carbohydrate chain. So what do we call a carbohydrate attached to a protein? Glyco for sugar, protein for protein, it's a glycoprotein. These are super important for things like cell-to-cell recognition. Now, what about molecule 2? It's that little round part of the phospholipid. It loves water, so it faces outwards. It's the hydrophilic phosphate head. So we're looking for glycoprotein and phosphate head. That's option C.
Okay, here comes a calculation question. I know, sometimes the numbers can look a bit scary, but don't panic. This is just about plugging numbers into the right formulas. We'll take it step by step.
Okay, let's crunch the numbers. First up, surface area. The formula is 2 times length times width plus length times height plus width times height. So that's 2 times 16 plus 24 plus 6. That gives us a total surface area of 92 square micrometers. Easy enough. Now for volume. That's even simpler. Length times width times height. 8 times 2 times 3 equals 48 cubic micrometers. So our ratio is 92 to 48. Now we just need to simplify that. If we divide both sides by 4, we get 23 to 12. Okay, part 1 done. Now for the second part. What happens if the cell gets wider? Well, here's a massive shortcut for you. Remember this rule. As any object gets bigger, its surface area to volume ratio always goes down. You don't even need to do the new calculation. You just know it decreases. So the ratio is 23 to 12, and it decreases. That means C is our answer.
Question 18. Osmosis and water potential. This topic can seem confusing, but honestly, it all boils down to one simple golden rule. If you remember this one thing, you'll get these questions right every time.
So here it is, the golden rule. Water always moves from a region of higher water potential to a region of lower water potential. Now don't let the negative numbers fool you. Think of it like a temperature scale below 0. Is negative 22 higher or lower than negative 40? It's higher, right? It's less negative. So just imagine water is lazy and always rolls downhill, from a higher number, like negative 22, down to lower numbers like negative 33 and negative 40. And which diagram shows the arrows pointing from high to low? It's A. Water is moving out of the cell, down the gradient. That's it.
Now we've got an investigation into beetroot permeability. Let's put on our detective hats and examine the evidence in this table. Let's start with option B. Treatment, ethanol. Observation, it turns red. Explanation, lipids dissolved. Does that make sense? Absolutely. Ethanol is an organic solvent. It will totally dissolve the fatty phospholipid bilayer of the cell membranes, causing all that red pigment to leak out. So B is looking like a very strong suspect. But let's check the others to be sure. Look at D. Treatment, hot water at 80 degrees. Observation, red. Explanation, lipids dissolved. Now the observation is right. Hot water will damage the membrane and make it leak. But is the explanation right? Not quite. The main thing that extreme heat does is denature the proteins in the membrane, which then makes it leaky. So the explanation is off. That confirms it. B is our answer, the only one where both the observation and the explanation are spot on.
And here we are, our last question, number 20. We've got a microscope image of a cell doing its thing during mitosis. So, first step, what stage are we looking at? Well, look closely. You can see the chromosomes all lined up neatly in a single line right down the middle, at the cell's equator. That's the classic sign of metaphase. Okay, but the question asks what happens next. So what comes after metaphase? Anaphase. And what's the big event in anaphase? The sister chromatids get pulled apart. They're no longer attached at the centromere. They're moving to opposite ends of the cell. And what about the nuclear membrane? Well, that disappeared way back in prophase, and it's not coming back until telophase. So during anaphase, the chromatids are separate, and there's no nuclear membrane. Option A describes that situation perfectly.
And that's a wrap on our 10 questions. So what's the big takeaway here? Think of this as your exam technique checklist. First, always hunt for those keywords in the question. Second, don't be afraid to use the process of elimination. Cross out the options you know are wrong. Third, try to connect every question back to a big idea, a core biological principle. And finally, when you see a calculation, just take a deep breath and write out every single step. Trust me, doing these things will make a huge difference.
Now, I've got a question for you. Out of everything we just covered, which concept was the trickiest for you? Was it the enzyme graphs, the surface area to volume stuff? Let me know down in the comments. Seriously, tell me what you found tough, because I might just turn the most popular answer into a full, deep dive explainer.
Awesome job working through all of that with me today here on The Explainer. Hey, if this helped you out, do me a huge favor and share, like, and subscribe. And for those daily revision hits, you can find Mr. Chi on Instagram, TikTok, Facebook, YouTube, pretty much everywhere. Keep putting in that hard work. It really does pay off. I'll catch you in the next one.